Okay, thank you. Hello, everybody. My name is Irit. I'm from the Israeli node. We are actually a mixed node. We have around seven centers. I'm just going to focus on ours. We are a small university, Bar Ilan University. It is located in the center of the country, near Tel Aviv. You can see the beautiful coast of the city. I think it might be the first time for our node to participate in this meeting, so I'm very excited and honored to be here. So let's start. Okay, so just a few words about our unit. We have a small light microscopy unit. I'm the manager with just 2 staff members and 2 students that assist us. We have a wide variety of instruments ranging from basic wide field light microscopy to confocals, spinning disk confocals, laser scanning confocals, and also super resolution STED. We also have a high content screening system, which I'm going to focus in this talk, and we provide some services for image analysis for students, for the researchers. Okay. So, yes. So what is high content screening? It's actually a combination between 2 classic methods, the high throughput screening and the high content imaging. High throughput screening is usually some biochemical assay that is done very, very fast on a large number of plates. It gives usually just one readout parameter that we measure and gives an average the signal over the entire well. So we don't have the special resolution, just very, very powering the statistics. The high content imaging, as you all know, is based on microscopy. So it has a very high resolution. We could get up to the single cell, and it can measure many, many parameters in very high resolution and in special resolution. But the problem is that it's pretty slow, so we can't get so many perturbations, and it is usually much lower in the statistical power. Okay, so the combination between them is the high content screening system. We acquired in our unit the Opera Phoenix Plus by Revity. I think it's the most advanced high content screening now present in the market. It is based on a fast acquiring spinning disk confocal, which can acquire many images quite fast, but in high resolution. We use the objective water objectives that are very good. We have 2 cameras, so it is faster. It can image in parallel, and it scans the plates automatically. We have this robotic arm. Oh, I'm sorry. You can see this robotic arm that can take the plates in and out of the incubator or the stands. These and then it acquires. And the most powerful thing about this system is it's the analysis capabilities. It is on a software for analysis, and I will show you some example of what we do with this. Okay, so the first study I'm going to show you is done by the Dr. Moran Dvela-Levit and her student Marquis. They study protein trafficking, and especially its engagement in kidney diseases. So it is known that 1 out of 3 of all kidney diseases arise from secretory protein trafficking defects. Okay, so they study the gene renin and see that if you mutate in different places along the gene in different segments, you can get different kind of disease in different severity. And most importantly, these will be the mutants will be accumulated in different compartment or organelles in the cell that is associated with the specific defect in trafficking that this mutant has. Okay, so what they designed is a set of 4 different cell types, the wild type and 3 mutants. Each mutant is mutated in a different place along the gene, will be accumulated in a different organelle and give a different kind of disease. So you see these 4 cell types indeed accumulate in different places in the cell. It can be in the ER, pre-ER, post-ER, Golgi, lysosome, and more. So now that they have established this model, they want to see how it works, the mechanism, do some screen, drug screen, genetic screen. Okay, so what was the challenge? Well, we want to see how we can make it more efficient. We need to screen as many as possible perturbations with the ability to label the retention organelle of each of the cell population. As each cell type, the mutation will be in a different place, in a different organelle. What are the difficulties? First of all, we have, for starts, 4 different cell types. And each of these cell type is associated with a different organelle. And we want to do many perturbations, such as check some stressors, do some drug screens, genetic screen, and you see it can get to a lot and lots of plates. Okay, so how can we make it more efficient, even with this high content screening system? We introduced a method which we call cell barcoding. What we do is we created these cell lines. Each one is a different mutant, and each was expressing a marker in a different color and in a different organelle in the cell. For example, we have the wild type that will be expressing red in the lysosome, okay? Another will be in the Golgi or the ER or in the cytosol corresponding to where it should be. Okay, so now that we have all these 4 cell types, we can pull them together into one well. Okay, so we have one plate with 4 different cell types. Now we can treat them together, we can monitor them together, we can later analyze them in one plate. So we increased the high throughputness by 4. How it looks? Well, this is one field with all 4 cell types. You see here, this is the wild type cell. We have yellow in the lysosome. This one is the signal peptide mutant, which have blue in the cytosol. And we also have 2 in green. First one is the mature mutant, which is green in the ER. And the second one is the pro segment, which is green in the Golgi. Okay. So now that we are, treating this entire well, now we need to separate them because we want to get each of these cell types analyzed separately, individually. So what we do now is use the image analysis of the software, and we segment them according to the color. So we have the blue, the yellow, and the green, with 2 mutants. I just forgot to mention before, we used only 3 channels because we want to leave 1 channel free for any other analysis that we want to do downstream, such as viability, toxicity. We want to stain for the renin, for the mutant localization. Okay, so we just need to use 3 colors. So we have these with 2 colors and 1 color and 2 sets of cells. And in order to distinguish between them, we used a machine learning module of the software that check any other parameters such as texture, location, intensity, and more to distinguish between these 2 cell types. As you see, they're located in a different place, so it shouldn't be that difficult. Okay, so now we analyze everything and we can continue for further downstream analysis of anything else we would like to do to distinguish between these cell types. Okay, so what's next? Now we can add more complexity to the system. We can add another, we are now planning to add another marker, an NLS marker, to mark the nuclei of the cell, okay? And now we can have one cell that will have an expression in one color in the organelle and another color in the nuclei, and we can create more combination. So now we can monitor many more variants of the gene in parallel, not only 4 but much more. Okay, so just to sum this up, we see that cell barcoding will increase eye throughput by 4, and in the future even more. Marking the retention organelle is biologically significant, okay? It's not just that we marked in a specific color, we also marked the specific organelle that can help us understand the mechanism of how it works, how the mutation and how the protein trafficking is defected. And the image analysis will allow us for subpopulation separation and to facilitate downstream quantification of each of the cell types. And lastly, all variants are pulled together and treated simultaneously, and that will minimize treatment variance. Okay, so I will go on for the next study. This is totally different. It is done by Professor Yaron Shaftal and his student Ilan. They study mRNA dynamics in living cells. So what we are doing in this is actually, I think it's unique. We integrated the analysis into the acquisition workflow in order to improve the acquisition efficiency. So I'll explain more. The goal was to image fast temporal accumulation. I think I will just, okay. This is the movie. We image fast temporal accumulation of different proteins at the site of RNA transcription in single cell in high resolution. I mean that we image the nuclei, okay? And we see inside the transcription factor if there is any accumulation of other protein that we want to check. What are the difficulties? First of all, we need to do it in very high resolution, single cell. We need to use a confocal, of course. Second of all, we need to capture as many cells as possible. But the problem is that we transfected these cells with 3 plasmids, and the transfection rate is not so high, and so some cells will not get all 3 colors. And also the cell density is not really very high. And of course, we need to image this as fast as possible to capture the dynamics of the protein in and out of the transcription site. So how do we do that? In conventional method, we just use to randomly select fields. Some did have the positive cells, some were totally empty, didn't have any cells, and in the end, we got maybe 10% efficiency. That means that most of the fields that we acquired were just useless. They didn't have any cells, or they have cells that weren't transfected with all 3 colors. So in the system, we can now use a mode that is called PreciScan. It is actually composed out of 3 steps done sequentially, one after the other. The first one is a super quick pre-scan that will scan the entire well, most of the well, in 10x magnification, all 3 channels in wide field mode. This is done very quickly and gets most of the cells. The second one is very important. It's an intermediate analysis step. Now, the analysis of the software will choose only cells, you can see in green, I'm sorry, only cells that has all 3 colors, okay? And in red, you see the cells that are not important, okay? And then it will determine the layout for the 60x rescan, okay, the final scan, and we'll get up to 36 fields that will be imaged. And the last step will be a rescan. Now we're going to start the time lapse. It will be in confocal mode, in 63x magnification, all three colors. We image 2 wells, 2 minutes intervals, up to 2 hours. Okay, so you see all of these steps were done automatically and sequentially. So we just push the start and it's starting, it's taking maybe 2 or 3 minutes and the time lapse will start. And the most important thing that we increase the efficiency of this whole process by to 100%. 10 times more than what we used to have. So you see that each one of the fields now will have at least one triple positive cell that we can further analyze for the accumulation of this protein. Okay, so what we did is to introduce an analysis step to the workflow to increase efficiency of this whole process to 100%. Okay, this is it. Okay, you can just look at the transcription sites and you see this accumulation of protein inside the transcription sites. Okay, so thank you. I want to thanks Euro-BioImaging for the support and for letting me the opportunity to talk to you today and for all of you to listen. Thank you.